So you need to be very careful, okay, and ensure that you don't invite any contamination at the time of spreading, okay. And we have antibiotics which is already amended, mixed with the media, but still you have to take all the precautions. So he also put it, the representative samples were plated out on antibiotic free media also, okay. That will tell you whether the cells are still viable and whether something has happened. So that is an indication that all is well with your experiment, okay. So this experiment will take another 2 days. On Saturday you just get into the incubator, get into your plates and see whether the colonies are coming up, and if colonies are coming up, you take the count and see how many colonies per plate is appearing. So end of the day, how many colonies you really expect? You want only one, right? You need only one colony, that is a genuine trans conjugate, okay. So but we always keep a population. Expectation is not only one, at least a sufficient population, so that you can take the best among them. If it is only one, there is no choice, right? If you are ending up in only one colony, you make sure that you repeat that experiment. All right. So at least you need some population. So then only you can say that the experiment is successful, okay. On this experiment, I think many of you might have done it for the first time or might have heard for the first time. If you have any quick interactions, you can ask. Anybody? The whole experiment is sitting on a single point, that is a neutral integration, isn't it? It's a neutral integration, right? You are adding an additional gene, a coding gene in bacterial genome, okay. So how do you call, can we term this particular event as a can we name it, this event? Can we name this event, integration of a specific gene in bacterial genome? Pardon? Site-directed mutagenesis. You are not precisely correct. We can call it as a gain of function mutation, because mutation is always having a negative connotation. When we talk about mutation, we always talk about something, in the by and large. But here, this is mutation only, but it is as gain of function mutation, because the organism. Let us see the colony first. First the baby should come, then only we can think of other celebrations, isn't it? Huh? Yeah, it's a kind of generic name. Trans conjugate means you are right technically, because you didn't perform any conjugation. But whether it is a triparental mating based conjugation or electroporation based transformation, end of the day, the final product is same only, right? Nothing wrong in calling trans conjugates, but yes, precisely if you want to call it as, you can call it as genetically transformed bacteria, because you did transformation, okay. So genetic transformation and genetically transformed bacteria and trans conjugates, it is functionally both are same. In both the cases, you are going to see expression of the inserted gene, right? In this case, it is a gene which is coding for green fluorescent protein, okay. So and the utility of the whole setup we already discussed in length. You can use this transformed bacteria for several other experiments. If somebody wants to know if it is a kind of biocontrol agent, many of you are working. If somebody is working on endophytic microorganisms, and if you want to give a proof that when I inoculate this bacteria in rhizosphere, and after a couple of weeks you are claiming that it is entering into the plant system, you can use this transformed bacteria for those experiments. And suppose if I want to know, I inoculate this bacteria on seed, and after a few weeks I want to see whether it is really entering into the plant system. It may be the root or stem or leaf or even further any other plant pot. You can track it. Basically it is a tracking tool. You can track the bacteria. So what you need to do is you collect the material, plant tissue, right, and go for fluorescent microscopy. After performing the fixation of the tissues in the appropriate chemical compounds. Okay, since somebody asked this, what is the utility of this particular system, it's quite easy. You take out the leaf or stem or any plant pot where you want to see whether the seed inoculated bacteria or soil inoculated bacteria is entering into the plant system and they are stably establishing their colony. So what you have to do, you take out those material, make a thin slice of those tissue, then you fix it in 4% paraformaldehyde. Okay, it's very simple. You make 4% paraformaldehyde, then it's simple. You prepare that 4% paraformaldehyde and take a slice of your tissue, just put it at, dip it in that 4% paraformaldehyde and just incubate overnight at 4 degrees. Okay, so what happens is, the trickiest part here is the sample preparation has to be done very carefully. Suppose if you want to claim that your organism is colonizing a specific niche within the plant, right? So your the very act of making the slice should not disturb the bacterial cells from its original place to another site within the endogenous region. So for that, that's a trick. See how to do many number of samples. You have to use very sharp blade or surgical blades and make a thin slice, rapid cuts, and just quickly you put it in 4% paraformaldehyde. So what is happening in 4% paraformaldehyde? The cells are fixed, right? Where it was there originally, it is fixed. Okay, then you rinse your tissues, tissue material, then just mount it on the glass slide and directly observe under fluorescent microscope. You don't have to do any other staining at all, right? So directly take those tissues on a glass slide and observe either under fluorescent microscope or confocal laser scanning microscope. Fluorescent microscope may be upright or inverted, whatever you have, and start looking for those cells which is giving you the fluorescent signals. Okay, so it is very comfortable if you have, you can do a lot of experiments if you have the genetically transformed bacteria which expresses the reporter gene. Is it clear to all of you? Right. So this is only a 10 days program. I don't think we can demonstrate the entire experimental setup. Let us see whether you are getting colony in the first place. If colonies are coming, then somebody was asking the confirmation, PCR based confirmation for insert. I already told you need to use 2 different primers. One primer is complementing the genomic DNA, the genomic region of the bacteria, and another one is complementing the plasmid, somewhere in the plasmid sequence, right? So this is a very unique PCR where it is going to give you results only when there are inserts. Free plasmids will not give you any amplicon. Is that clear? Right? So how many of you are going to do this transformation after this training? 1, 2, 3, 4, 5, right? Okay, let us see. Okay, any more doubts on this? Everything is clear? Okay, then Saturday we'll come to know whether it was done properly or not. Okay, we'll. Okay then, so it's already 10:10 now. We'll get into the first lecture which is scheduled this morning. Welcome, Deepa. You're okay? Good. Okay. We have kept one important practical this afternoon, followed by another practical. These two practicals are interlinked. Today's practical is all about isolation of RNA from microbial samples. Okay. This practical you do it in the PG lab this afternoon. There may not be any time today to carry forward this experiment into qPCR that we will do tomorrow. Okay, but before that you need to know what is real-time PCR. How many of you had experience on real-time PCR? 2. I think 2 hands are raising very fast. 3. Okay. So I'm glad to know that at least some of you know this real-time PCR. That means majority of the students are they may be new to this experiment, correct? Or you might have heard about it, isn't it? The purpose of this lecture is just to let you know what is this real-time PCR and what are all the variants of this real-time PCR, and wherein we can use this real-time PCR in plant pathology and microbiology. Okay. So PCR all of us have done, correct? All 25 of you have done this PCR here, isn't it? Isn't it? So real-time PCR we will do tomorrow. So application of real-time PCR for pathogen quantitation and gene expression. I put it in the title deliberately because these two are the major applications of this real-time PCR in our area of research, right? So I just wanted to change it, so I just changed the title. It's very simple. Application of real-time PCR in plant pathology and microbiology. Okay, I'll take you the a small tour of the genesis of this real-time PCR technique. It's very interesting. So this is the real-time PCR tool is a relatively new tool, started evolving after 1990. Okay. No problem. no problem. If you look at the history of PCR, right? All of us know that the PCR technique was evolved. The techniques and the associated tools, machines, it started coming into science after 1980s. Correct? I think it's well known to all of you. But the prototype concept and the prototypes, it started appearing in a small way in science in 1970s also. Okay, people started thinking about in vitro multiplication or amplification of nucleic acids. Concepts really started evolving in 1970s. The first report of replication of single stranded DNA from a template using synthetic primers and DNA polymerase. It traces its origin in 1970s. Okay, in 1976, the DNA polymerase, one of the best known enzymes in molecular biology, that was isolated from a thermophilic bacterium, that is Thermus aquaticus. So, that has happened in 1976. 1983, Kary Mullis, he invented the polymerase chain reaction. Okay. In 1985, the first machine came to science which automated the concept of Kary Mullis. Okay. So, before that people used to move from one temperature to another temperature just to expose the reaction cocktails. So, now in 1985 the machine started coming from different companies which automated the PCR. 1989, of course, the Science, one of the famous journal, it rated this DNA Taq polymerase as molecule of the year, right. So, because they believed that this enzyme would change the science, the biology and molecular biology once for all. So, aptly they called this enzyme as molecule of the year. Okay. So, whatever they believed or whatever they expected, that has happened in after 1989. It changed the biology once for all. In 1993, Kary Mullis was awarded Nobel Prize for his invention, the polymerase chain reaction. Okay. Of course, virtually scientists in all branches, whether it is a biology or it is medicine, forensic, diagnostic, plant sciences, environmental sciences, in every field of biology, you cannot imagine a research program without PCR. We are so dependent on this tool for our day-to-day experiments. For some applications, the qualitative nucleic acid detection is sufficient. Okay. There are many applications, yes or no, right. The classical one is detection of pathogens, detection of microorganisms, right. That is the best way of telling the results, yes or no, isn't it? If amplification happens, expected amplicon comes. I hope all of you understand by making a statement amplicon. Amplicon means the multiplied fragment, right. This is a jargon, amplicon, the product which is coming after amplification. So, now the qualitative nucleic acid detection is sufficient in most of the cases, right. But there are experiments, right, especially in genomic science, more precisely in transcriptomic science. It is not the qualitative results are important. What is more important is the quantitative results. It is not what is more important, the what is getting amplified. What is more important is how much is it is getting amplified. Okay. The real time PCR, it can be used for both qualitative and quantitative analysis. Okay. For the students level, PCR is the one, a normal PCR. By making PCR, making a statement PCR, what I mean, a normal PCR, that is typically qualitative in nature, right. And the real time PCR, it has got the additional benefit, it can tell you not only what is getting amplified, but also how much of that amplicon is getting amplified. Okay. So, of course, let us see what is this. Much like the history of PCR that happened in 1980s to 1990s, the 10-year time point from 83 onwards up to 89, the science rated this molecule as molecule of the year. The real time PCR development, it took place after 1990s. Okay. Hikuchi in 1993. So, he recognized that the process of PCR could be monitored. We all know that in PCR what is happening? The nucleic acid is amplified, right. So, you wait for a couple of hours, right. The machine will tell you all cycles are completed. Now, you open the machine and take out your product, and you wait for another 2 or 3 hours to know whether the amplification was successful or not. Okay. That means, from the start of the experiment, the end of your electrophoresis, it may be around 4 to 6 hours time gap is there, just to know whether the amplification took place or not. Okay. But in real time PCR, right from the first cycle, right, you are having an option to see whether the amplification is happening or not. Okay. That is the beauty of it. So, what he recognized, the process of PCR could be monitored just by adding some fluorescent dyes or fluorescent label that binds to the accumulating PCR product. It was a concept. So, he told if you add some fluorescent dye to the amplifying product, the entire process of your amplification can be tracked, can be monitored. So, as the concentration of PCR product increases, the intensity of the fluorescent signal increases. Okay. So, he hypothesized that it is possible to track the process of amplification. So, he suggested to use ethidium bromide. We know that ethidium bromide binds with DNA. Okay. So, ethidium bromide bound DNA can be visualized. So, it is not surprising. The only thing is he had some out of box thinking. So, he proposed that. This particular discovery paved the way for modern quantitative real time PCR. This is the genesis of real time PCR. Okay. When ethidium bromide is bound to double stranded DNA and excited by UV light, it fluoresces. Okay. You might have seen gel what is called as gel dock system. Simple, right? So, if you do not add ethidium bromide, you forgot, then what happens? You do not see anything, right? So, then you regret, oh, I forgot to add, isn't it? In 1996, there is another invention that complemented the concept of Hikuchi's, ethidium bromide based tracking system of amplification, the Tachman detection method. We will have several slides on that, we will discuss that. In 1996, the Tachman detection method used instead of ethidium bromide for real time detection of PCR. Okay. In 1996, a company called ABI, the era of PCR, 1990s, the era of real time PCR. Okay. In 1996, ABI, there is a Applied Biosystems, there is a company. This company launched their product that automated the real time PCR, and that machine was called as sequence detection system. Okay. That machine is ABI 7700. So, this is a machine, how it looks, it is a huge machine. Okay. Now, what is the fundamental difference between this conventional PCR and real time PCR? I already highlighted. We are more bothered about what is amplified in conventional PCR, but in the case of real time PCR, we are bothered about how much it is amplified. Okay. So, this how much is addressed by adding some additives in the PCR. Okay. Then you have a system of tracking the additive, how this additive behaves. Additive here is a fluorescent dye. Okay. Then you have a system of track how much of this dye is increasing. Let us see how it is really done. This is a specialized technique that allows you to monitor the reaction in real time. Okay. What is happening in the PCR is monitored in the real time. The webcasting is all about that only, isn't it? It is a real time monitoring of events, right. Of course, the applications are plenty, let us not get there. There are virtually every branch of biology we have an application of PCR and an application of real time PCR. Okay. We have application directly in plant pathology, microbiology, pathogen quantitation, microbial load estimation. So, and the list goes and goes and goes. Yeah, you may call it as a quantitative real time PCR, also called as qPCR, and a method of polymerase chain reaction that allows to follow in real time the amplification of target. Okay. Since the technology allows you to track the events in a real time basis, this is termed as real time PCR. Sorry. The real time PCR is a technique in which fluoroprobes bind to specific target region of amplicons to produce fluorescence during PCR. Okay. So, maybe the coming slides will enlighten you how this is exactly done. The fluorescence measured in real time is detected in PCR cycles or PCR cycler with the inbuilt fluorometer, right. So, it is a normal PCR mixture only plus one additive, right. The normal PCR machine does the PCR function. This additive helps you to track what is really going on in PCR mixture. Okay. Let us see how this machine looks like. Real time PCR is nothing but a PCR machine combined with spectrofluorometer. There are two machines in the single machine. There is a PCR machine which is combined with spectrofluorometer. Okay. This is the spectrofluorometer which is kept on top of the PCR machine, and this is this is the PCR machine. Okay. It is something like you have a PCR reaction which is going on and somebody is monitoring it, is sitting on the top. Okay. So, it is a combination of a PCR machine plus spectrofluorometer. And of course, the typical fluorescent detection, only it is exploited here. There is an emission, there is excitation, there is an emission wavelength, and that is captured by spectrofluorometer. This is a typical conventional PCR. You have already done it, right? You have a primer, you have template, DNA, and you have DNTPs and other additives, and of course, you have the most important enzyme, that is DNA polymerase. When it comes to real time PCR, you have everything, and you have one extra thing, that is detector. The detector is the one which is going to bind with the amplicon. After binding, the amplicon gives you some fluorescence, and that fluorescence is captured or monitored, right, by the spectrofluorometer which is sitting on top of the machine, right. So, detector. So, you know all other ingredients, right. DNA is from your side, primer of course you only designed it, and these chemicals are purchased, right. So, now the question is, what is this detector? Who is going to supply you, and how to procure that? So, right now we have two major classes of detectors. One class of detector, what is called as a fluorescent dye, also called as non-specific detection. Okay. The fluorescent dye based real time PCR is one option. Another option is specific detection, that is through detective probes. Is it clear? Two options. Option number one is a fluorescent dye based detection. Another one is detective probe based detection. Okay. So, the first one by and large we call it as a non-specific detection, and second one we call it as a specific detection, that we will discuss in the coming slides. What exactly is the difference? This is also called as dye based quantitative real time PCR, and this one is probe based quantitative real time PCR. Is it clear to everybody? Let us see one by one. What is this dye based and what is this probe based? Dye based quantitative real time PCR, right. It was the original concept of Hikuchi. It already told, he used ethidium bromide. Okay, that was replaced by other safer dyes in the later years, right. Let us see how exactly it works. Dyes, there are a range of dyes are there which can bind with DNA, okay, which can have a specific affinity towards DNA. So, cyber green, right, that intercalates into double stranded DNA, okay, or more convenient to have on hand, less expensive than probes. The dye based non-specific real time PCR is one of the easiest real time PCR to perform, no complications at all. You have PCR mix plus one extra chemical, that is the detector. All you need to have is a real time PCR machine in your lab. Okay. You can you can do this real time PCR. Right. Of course, I already highlighted in the last slide. The flip side of this dye based real time PCR is it is non-specific. Why it is non-specific? You will understand. The binding of this dye on DNA is a general phenomenon. Okay, nothing to do with the sequence specificity or sequence, the intrinsic sequence, what is there. Any DNA, this dye will go and bind. The only thing is it need double stranded DNA. Okay. Let us see how it works actually. This is some options. Okay. Do not think it is only cyber green all the time. You have different chemicals are available which can bind with this DNA, starting from cyber green 1, cyber green 2, Yuga green, LC green, Pibo, Ypro, Cyto family, and there are many. Right. This is the principle behind this cyber green based real time PCR. Okay. So, just recall the mixture of real time PCR chemicals. What did I tell you? You have all components of PCR plus the detector. Okay. So, you mix everything time 0, what is inside now? You added everything, you have template, you added primer, DNTPs, magnesium, buffer, right, maybe other additives, sometimes people use BSA, DMSO, you add for some difficult amplification region, we add all these things, plus the dye, right. So, what happens in the first denaturation cycle, what happens? Denaturation. Okay. After denaturation, what happens? Primer binding. Okay. After that, what happens? Extension. Right. So, basically what is happening here? A double stranded DNA is converted into 2 single stranded DNA. Correct. These 2 single stranded DNA is converted into double stranded DNA. Okay. Remember, this is happening when you start your thermal cycling. Correct. So, now you put the template. How much template you put? We already discussed. Some applications we put 50 nanograms, 100 nanograms. I suggested 200 nanograms, right. So, along with that you have dyes also. Okay. So, what is happening is when you set up PCR, the cycle, the repeated cycling, add up further double stranded DNA from the template. So, there is a progressive increase of double stranded DNA. So, during that step what happens? In a solution, unbound dye exhibits very little fluorescence. Why? Because the template is so low. How much template you put? Maybe 50 nanograms or 100 nanograms, right. So, in principle what happens here? Double stranded DNA will bind with dye, but that basal fluorescence is usually not detectable. Try to understand this. It is not usually detectable. Okay. So, the dye what was added in the mixture, it is a unbound dye which exhibits very little fluorescence, or if there is a little binding takes place, that is usually not detectable. Okay. Now, the cyber green binds to the minor groove of double stranded DNA helix. when it happens? When it becomes 2 single stranded, primer binding and extension. So, at that stage what happens? The cybergreen has got a specific affinity towards the minor groove of double stranded DNA, correct? So, it goes and binds there, right. So, as the double stranded DNAs are formed after extension, the unbound dye from the solution goes and binds in the minor groove, right. After annealing of the primer, the dye molecules can bind to the double stranded DNA. So, the unbound dye what you added in the mixture, it goes and binds whenever there is a formation of double stranded DNA. Is it clear to all of you? Now, the DNA binding results in dramatic increase in cybergreen molecules to emit light upon excitation. So, every time when there is a double stranded DNA forms after annealing, the free floating dyes goes and binds in the minor groove of the double stranded DNA. So, that is what depicted here. So, initially there was nothing and it started now, and after one round of amplification, the dyes are already bound with the minor groove of double stranded DNA. Okay. Let us see what exactly happens now. The progressive increase in the fluorescence, progressive increase in fluorescence as the amplicon accumulates, isn't it? The intensity of fluorescence is measured by spectrophotometer. Okay. So, this is what exactly happens. So, you can see the number of bound dye which increases with time. Okay. This is clear to all of you. So, as amplification increases, the progenies or the daughter double stranded DNA what is amplified in PCR, it goes on adding the dye in the minor grooves. Okay. So, what is the advantage of this now? You can track what is really going on. So, initially there may not be any fluorescence because machine is unable to see what is going on there. Then after sufficient number of the double stranded DNA accumulation, the quantum of double stranded DNA what is accumulated there is equivalent to the quantum of fluorescence what is coming out of the double stranded DNA, okay, the fluorescence. The fluorescence intensity is captured by spectrophotometer. Okay. So, this is in nutshell how this dye based real time PCR usually operates, right. So, what is the advantage here, the big advantage? You do not have to do anything, you have to buy only this dye and incorporate in your normal PCR mixture. If you have a real time PCR machine, that means a PCR machine which is attached with a spectrophotometer, then your real time PCR experiment can be performed now. Okay. Very easy, relatively cheaper, very easy to handle, very sensitive, no doubt, right, and of course, very effective. What is the disadvantage? By chance your primer is a wrong primer. For example, I am telling you, the primer is not very specific, correct? Possible, na? Instead of targeting flea-C gene, it goes and amplifies something else. In real time PCR, what happens? It does its job, no matter what is amplified. That is a problem. So, that is a disadvantage of this particular technique. That is what in the previous slides I told. The dye based real time PCR is not specific. Okay. This is clear to all of you, all the students. Anybody is having any doubts, I can repeat. Okay. Now we will move on to the probe based quantitative real time PCR. Okay. How many of you have done this? I am sure none, correct? The dye based one is very popular, everybody uses it. Okay. They claim that my primer is the best one in the world, right? So, I do not need probe based real time PCR. Yeah, there is a logic, strong logic. If you claim that your primer is the best one, it amplifies only that gene what I wanted to do in my thesis, right? Then you be comfortable with cyber green. Okay. But since this is one of the finest, extremely sensitive, highly specific technique, let us know what it is. Okay, this is probe based quantitative real time PCR. Okay. In fact, it was invented right in 1996 itself, and people started proposing this idea. Okay, the probe based. The probe based qPCR relies on sequence specific detection of desirable PCR product. Okay. We will elaborate that in the coming slide. Unlike cyber green based qPCR method, which detect all double stranded DNA, the probe based qPCR utilizes fluorescent labeled target specific probe. Try to understand that. Fluorescent labeled target specific probes. Okay. At this point of time, you please confuse with the primer. Okay. We will remove that confusion after some time. Right. What do you mean by target specific probe? It is nothing but primers only, right? We are only telling in a different name, correct? One point. So, the principle here is we are going to use fluorescent labeled target specific probes. I am not calling it as target specific primers. That is what the choice of word is very important here. Okay. We will come to know what is the difference between these two, the target specific probe and the primer, because every PCR you need primer, correct? Without primer you cannot do PCR, isn't it? Without primer and template there is no PCR. Nah. So, let us see what exactly is the difference between this fluorescently labeled target specific probe and the primer what you designed for your experiment. Okay. Let us discuss that. And what happens? The probe resulting in increased specificity and sensitivity. Right. Much like the dyes, you have different options. I told different of cyber green 1, cyber green 2, Eva green, so many things. Much similar to that, you have different kind of probes also. Right. You have a Tachman probe, the most popular one. We have molecular beacons, we have scorpion primers, hybridization probes, and again the list goes and goes. Okay. So, pay attention here, it is very important. What I put here in the top, R and Q, okay. That is what I told, fluorescently labeled target specific probe. Okay. Just have a look, there is a probe, right, and somewhere on the way this probe is disintegrated, correct? There is a probe, the probe has got two components, there is one written as R, another one written as Q. Somewhere down the lane you are seeing these two are separated from each other. Okay. Keep in your mind. Now, you go through the statements. When the fluorescence of the reporter, this is reporter, is quenched. What do you mean by quenching? Temporarily silenced or suppressed, whatever may be the language, right? So, this is most important point. The fluorescence of the reporter is quenched. quenched due to the proximity to the quencher. This is the most important one. If you do not understand this, you will not understand the probe based real time PCR. The probe has got two components. One is the reporter, another one is quencher. When these two are close to each other, separated by few nucleotide bases, right? Much similar to the primer only. The only thing is it is decorated with two chemical molecules. One is called as reporter, another one is called as quencher. So, this quencher, what it does? It does not allow the reporter to give fluorescence or give light, switched off. Okay. When the probe hybridizes to the target, remember this probe is much similar to your primer. It goes and binds to the complementary sequence in the target. Is it clear to all of you? Anybody? Not clear. I am not talking about primer here. I am only talking about fluorescently labeled target specific probes. This is having reporter and quencher. This behaves much like primer, right? It goes and bind. Okay. Is it clear? So, when these two are together, the reporter is taking rest, okay, silenced. If you remove the quencher by some mechanism, this reporter will give you fluorescence. Now, how this is done? Let us see. The polymerase enzyme, which is adding the nucleotide after primer binding, this has got another activity. How many of you know this? 5' to 3' exonuclease activity. Okay. This activity is very, very important for this probe based real time PCR. Okay. Double stranded specific 5' to 3' exonuclease activity of TAC, it cleaves off the report, rather it disintegrate the probe. Okay. After disintegration, what happens? Reports are separate, quencher separate, the few nucleotides also removed, correct? The reporter is separated from the quencher. When they are away from each other, the reporter gives you fluorescence. Okay. So, fluorescent signal. The signal is proportional to the amount of amplified product in the sample. Let us see how it happens. Next slide. The mechanism of TACMan based real time PCR. Right. This is your probe, fluorescently labeled target specific probe. It is having the reporter, also called as fluorophore. Is it clear? And you have a quencher. Right. So, now what happens? When you start your PCR, what is the first step? Denaturation. Double stranded DNA will become 2 single stranded DNA, correct? Now, what happens? Usually what happens? Primer will go and bind, but here primer also will go and bind. Parallelly, the probe also will go and bind. Is it clear? This you should understand. Okay. What is probe after all? It is complementing to the target. Functionally, it is equivalent to the primer. The only thing is we have decorated it by adding 2 molecules. One is the reporter, also called as fluorophore, and also the quencher to silence it as long as they are together. Is it clear? Now, see the first step, denaturation, right? Imagine a solution. You have double stranded DNA, you have primers, you have nucleotides, you have probes also, right? The moment it becomes 2 single stranded DNA, the reaction started. all molecules started moving here and there in a chaotic manner, correct? Probes and primers will go and bind wherever they have the complementarity, right? It's a parallel. Remember, it's a parallel event. Okay, it's not one after another. It's a parallel. Okay. Now what happens? The first step of denaturation, it becomes 2 single stranded DNA. When the 2 single stranded DNA comes there, there comes the action started. All your primers and probes started moving towards the complementing region in the single stranded target. Okay. Now what happened here? I am only showing probe first. Okay. Now the fluorophore and quencher, it found the complementary sequence in the single stranded DNA, goes and binds there, right? It bound here now. Is it clear? You can imagine a primer also will come here. Yes, right. The primer also will go and bind. Where it binds? Where you design the primer, right? The only point you need to remember here is probe sequence and primer sequence should be different. You cannot use the same primer. Keep in your mind. Okay. Let's move on. What happens? 2 single stranded DNA. I am just showing one of the strands is bound with the probe, correct? And you may find that primer also somewhere sitting here. Yes, it is. It should be happening. Let's see exactly, right? So what happens? Annealing, right? The moment single stranded DNA opened up, probes found, you have your primer also found, bound. Now what happens? After primer binding, what happens? Extension. How this extension happens? Polymerase enzyme, right? When polymerase enzyme comes after primer binding, it started adding the nucleotides corresponding to the template. When A is there, T will come. When C is there, it will call G. When G is there, it will call C. When T is there, it will call G. It comes one by one. So remember, I already told the same enzyme has got 5' to 3' exonuclease activity. So it is adding a new basis. At the same time, it is removing whatever comes in its way, right? So what is coming in its way? Probe. Probe is having fluorophore and quencher. So it starts removing that. The primer binding on the complementary template. DNA polymerase, right? The green one. Now what happens? It started adding the nucleotides, right? Now, the moment it touches the fluorescently labeled target specific probe, it is going to remove whatever comes in its way, right? It removes the quencher, it removes the short fragment, and also it removes the fluorophore. Okay. Now what happens? 5' to 3' exonuclease activity of polymerase removes the fluorophore, right? And probe nucleotides, correct? And also the quencher. So only these 3 are there. Nucleotide can be any one of the 4 or mix of 4, whatever. We do not know what is that. Maybe a different proportion of A, T, C, G. It is cleaved off, right? Not only cleaved, it is disintegrated. Now what happens? Your quencher has come out. The fluorophore emits fluorescence after detachment from the quencher. So what I told, when quencher and fluorophore are together, separated by few bases, usually the fluorophore does not fluoresce. To make it fluoresce, you have to remove the quencher. The removal is done by the very same enzyme which is performing adding or extending the sequences. Okay. Let's see what exactly happens here. Now, enzyme, right? It removed this is, just imagine, this was previously an intact probe, right? After binding, it got disintegrated. So there is a quencher separate and reporter separate or fluorophore separate and other nucleotides, right? Every cycle this is repeated, right? So just imagine at the time of start of your real time PCR, you added probe together. Together you added, correct? It's a short, like much like your primer you added. There is a probe, there is a quencher, there is a reporter, nucleotides, and as the amplification progresses, it started adding, it started binding with the target and getting disintegrated after the primer binding and primer extension, right? This is what the scenario. So after the run, right, and before the run, what is the difference? Before the run, the probe was together. After the run, probe is disintegrated into fluorophore separate, quencher separate, and the nucleotides which connected both of them separated, right? And your primer has done the priming and annealing, extension. So you have a lot of double stranded DNA molecules, plus the fluorophore has accumulated in the PCR mixture. Okay. As the cycling continues, as the double stranded molecule accumulates, there is a progressive increase of fluorescence, right? 5 prime to 3 prime, 3 prime exonuclease activity of the polymerase removes the probe, nucleotide and quencher, and every amplification adds free fluorophores in the PCR mixture. That is the point. Every amplification step adds a free fluorophore which is not connected with quencher in the PCR mix. So what is the benefit of it? The entire mix gives a fluorescence. Initially it will be low. As the amplification increases, the intensity of fluorescence increases, and that is almost corresponding to the amplicon which is getting accumulated in the PCR mixture. And the intensity of fluorescent signal is captured by spectropluorometer. Remember, many times the confusion should not be there, whether it is cybergreen based or it is probe based. The machine is same. There is no separate machine for the probe based real time PCR. Machine is same because there is only one extra component sitting on the top and monitoring what is going on. Okay. Only thing is, in cybergreen, it captures the fluorescent signal which is coming from double stranded DNA. But in probe based, it is capturing the signal coming from the PCR mixture itself. Remember, double stranded DNA per se is not fluorescing here. Is it clear to all of you? Why? Any one of you? Why the double stranded DNA is not fluorescing here in probe based? I am asking you. Pardon? Can you give a mic to her? I would like to know. Why in probe based method double stranded DNA is not fluorescing? Say it loudly. Say it loudly, please. Quencher is separate. Very good. Is clear to all of you? Okay. What's the advantage? Extremely specific. Suppose your primer fails. Nah? It might help, I am telling you. So it is not the case. Primer should be very specific only, right? So the point here is, who will help you to design the probe? How the probe is made? Who will do it? Can we simply order and buy the probe? Somebody already did real-time PCR. They can answer. Just please give the mic. Ilakya is there. So who is designing primer, by the by? You are designing the primer, correct? Now the question is, who is designing the probe? Please, Ilakya. We have to design probe also. Okay, then who will manufacture the probe? Kavi has done. Let us see. Okay, rightly told. So that is extra step you may have to do it. So that's the reason why people are so comfortable with cyber green based qPCR, hassle free, right? But here what you have to do? You have to go for 2 times primer designing, correct? Probe designing is also much similar to primer designing only. Okay. So you order it, get the probe done, then you set up this. Okay. So for that reason, it's extremely specific, right? And of course, there are some disadvantages. Let's not discuss that here. So with that, I just conclude the first part of this real-time PCR lecture. Basically, I just wanted you to know the fundamental difference between dye-based real-time PCR and probe-based real-time PCR. I hope you all understood this. If not, please let us clear it here itself.
What should be the size nucleotide of the probe?
Much similar.
Same as the primer. Okay. But any range is there, like primer we should go up to?
See,
like
ultimately it need to be removed, and you have to give an opportunity for the enzyme to remove as quickly as possible.
As well as the annealing temperature also should be same.
So it takes all the criteria of primer designing, right? Isn't it? So the critical point here is it's a parallel event. I already told it's a parallel event. It's a process which is happening parallelly, right? One of the speaker told here, nanopore. It is adding 450 nucleotide per second. Just imagine what's happening there. Such a rapid, addition of nucleotides, right? So your question is slightly linked to that only. As short as possible, and it should not miss prime or miss complement with some other region. It takes all the criteria of your primer design. Okay.
So, sir, in one reaction, one probe only for the means 2 strands are there. Only to the specific one, it will go to the bind on the single strand. That one strand.
Yeah.
So for every event, there is a 50% of the fluorescence only will get, not the.
Doesn't matter, because end of the day, you want the free fluorophore in the PCR mix. That's it. That's the only thing.
But when we'll do the quantification, so we have to double the amount, that this much amount has.
See, it's not like that actually. See, after all, real-time PCR is real-time monitoring of what's happening in the PCR mixture. Okay, that you are able to see the original concept, whatever they told, the mechanism of monitoring the amplification process by using the dyes. Okay, so as and when one cycle is completed, you are going to see the free fluorophore which is coming out of that particular strand, right? So every amplification step, it is going to contribute a free dye molecule in the solution. So that is tracked. Okay.
Can't we infer the specificity of amplification and dye-based qPCR by analyzing the Melt curve?
Yeah, we can do. I am just coming to that. Yeah. We can do that. Yeah. Melt curve analysis, anyway, we have to do it. In few slides, I will show you. This is clear to all of you?
Sir, there is also possibility when it is annealing is there, means primer and probe are simultaneously binding, there is also chance that sometime primer may bind, but in that particular reason probe may not bind, but that product will get amplified. But we are, as you are seeing, that only fluorescence will get counted, but that product is already amplified, but that is not detected. So is it?
It might happen. We see that kind of mistakes might happen definitely, but what we anticipate is the binding specificity of your probe and the primer is nearly equivalent. So the short nucleotide sequence of the probe, I am sure such a probability, even if it is there, that is not going to hamper your experiment, right? Even if it is there, but I seriously doubt whether such a things can happen. That means primer is not binding, means your primer PCR will not work, right? The majority of the template are bound with the primer, then it is successful PCR, right? We don't expect all the template to be binding with the primer. One or two miss priming or no priming, it's okay, right? Anybody?
Suppose without RT-PCR, I'm going for normal PCR and quantifying using nanodrops. What is the significance of CQ value and normally going PCR quantifying and that endpoint analysis, and what is the significance between this?
Yeah, we will
CQ
come. You introduce another terminology, CQ values. I never talked about it so far. So we'll take that in few slides, and also more so in our practicals. We will be demonstrating how to convert the CQ values into copy number, right? We have a practical lined up tomorrow, that is absolute quantitation of target. So how to convert the CQ values into the copy number. So that's exclusive practical we have. We'll discuss that point. Okay, you wait till that point of time. My point here is just to throw some light on real-time PCR, so that you understand the basic concepts of the two different real-time PCR techniques, which is ruling the biology now. The more so the dye-based one. Okay, that is the primary focus of my lecture today. And in practical, we will clarify whatever doubts you have. How to analyze the data for absolute quantitation, how to analyze the data for relative quantitation, all those things we will do in the practical class. Okay. If these two concepts are clear to all of you, just move on to a few more points. That is all about reverse transcription. Many times in literature you will find RT-PCR. It's always confusing in biology, right? It's a real-time PCR or reverse transcriptase PCR, right? So much of confusion is there in literature. Sometimes people call qRT-PCR, right? Sometimes RT-PCR, sometimes qPCR, right? I think the most accepted terminology is qPCR now, right? We all agreed. Let us not confuse too much. We'll simply call it as qPCR, okay? qPCR may be for absolute quantitation, it may be for relative quantitation. That you have to specify in your manuscript. What for you are doing? qPCR. Okay. This is nothing but what is called reverse transcriptase PCR. This is more often we use in relative quantitation, especially gene expression analysis. Okay. Most of you might be doing in your routine thesis project. Okay. You might be knowing how to do that. So you have to go for RNA isolation, you have to convert into cDNA and use the cDNA as a template. That means it becomes reverse transcriptase PCR. If you use qPCR technique here, then it is again real-time PCR, right? So come to the mixture here. This is again I am showing the same thing here once again. The template here is a template derived from RNA. So today's practical is on that only. We will be demonstrating how to do RNA isolation, how to convert the RNA into cDNA, right? And this also very common, terminology you will find in literature, as one-step real-time PCR and two-step real-time PCR, right? So it's nothing. It's very simple. When you are working with RNAs, RNA cannot be directly amplified. You need to convert into cDNA, right? So this cDNA conversion of RNA into cDNA we do in one particular time,
now your cDNAs are ready. Then next day you take this cDNA and set up your real time PCR. It becomes 2 steps, correct. Now we simplified it. Why 2 days? You do everything at once. You take RNA, you put everything inside, right. You put reverse transcriptase enzyme, you put polymerase enzyme also. So the conversion of RNA to cDNA is done in the same tube where the amplification is going to be performed. So that is what is called as one step. Simple. So this is one step. You put everything here. You can see here reverse transcriptase enzyme, and you have DNA polymerase enzyme together. Both enzymes are together. Plus the rest all ingredients what is needed for converting cDNA, converting RNA into cDNA, and also the cDNA into the amplicon library or whatever you call. And that is a single step. In 2 step, again the RNA is handled separately, the cDNA is prepared separately, and the stock is already preserved somewhere, and as and when needed you just go to cDNA, take it out and set up the PCR separately. So you can see the reverse transcriptase is separate here, the rest all ingredients which is needed for doing the reverse transcription. Then the end product of this is cDNAs, and those cDNAs are used here along with the DNA polymerase, and the PCR is going to be performed. Is it clear? 1 step RT-PCR and 2 step RT-PCR. The one what we are going to demonstrate tomorrow here is single step RT-PCR. Okay. Requirements. What is needed? The primary requirement is you need to have a machine. If you don't have real time machine, you cannot do real time PCR. Correct? Isn't it? You mix everything and don't look at the tube, nothing will be seen. You need machine. Correct? So a real time PCR machine for running the reaction is absolute requirement. Okay. And of course template can be a DNA if it is absolute quantitation, environmental sample, or RNA, cDNA derived from RNA, mix anything what you handle. So you need a template, and you need primers if it is dye based. If it is probe based, you need probe also, isn't it? And yeah, it's probe based, right? And of course the DNTPs, and you have other ingredients what is needed for real time PCR, and of course the TAC enzyme and different kind of chemicals. Don't worry too much about it. Your only botheration is your primer and probe, if it is a probe based, or only primer, right? The rest all can be procured from one other sources. This is not be exactly the one what is displayed here. I just showed. There is no difference. It is much like the normal PCR. You have a denaturation, you have annealing, you have extension, final extension. That's a classical workflow what we normally do. Yeah, this I will introduce some terminologies. We will have a detailed discussion in the practical class. Okay, let us not have a very elaborate discussion here. This is a typical, the result of real time PCR. You see a kind of typical S-shaped curve. Okay, what is displayed here is the results of real time PCR. Okay, so and there are some parameters. This is called baseline, the initial phase, this exponential phase. Here it is called CT or CQ. Somebody was asking. This is the plate shape, and this is called threshold. We'll discuss this in the practical. What it really convey to us in terms of amplification. Okay. Baseline amplification plot, the initial cycle of PCR, there is a little change in the fluorescence signal, which I already told, right? The fluorescence molecules are very little, whether it is cyber green or it is a probe based. Initially, it is very, very less, right? Why it is very less? In the cyber green, the template quantity is so low, and whatever little fluorescence happens, because there are some minor grooves, cyber green go and bind there, and signal is so weak, the machine is unable to capture it. Okay, the fluorospectrophotometer is unable to capture it. In probe based, of course, it is in the bound state, the quencher is not allowing the fluorescence to happen. So, either way the fluorescence is not there. So, as the amplification started, the fluorescence comes and the fluorescent signal is captured, right. Then there will be a point, there is no more fluorescent accumulation, because there is a limitation, everything is a limiting factor here, right. There may not be sufficient template after this stage, there may not be even dye, right. So, saturation, right. So, curve does not go further, right. An increase in fluorescence above the baseline indicates accumulated PCR product. Okay. Threshold line, another terminology, point at which a reaction reaches a fluorescent intensity above the background. Okay. It is set in the exponential phase of the amplification for the most accurate reading. It is always somewhere in the midpoint of your exponential phase. Okay. Cycle threshold, CT or CQ, the cycle at which the sample reaches the threshold level. It varies from sample to sample and it is directly decided by the initial template concentration. Okay. And of course, by and large, the CQ value 40 and above considered to be failed PCR. Okay. This is melting curve, somebody was asking, right. In cyber green based real time PCR, fluorescent signal comes only when there is a double stranded DNA. Okay. There is always a melting point for double stranded DNA. If you subject your sample to that melting temperature, what happens? There is a sudden drop in fluorescence. Why? Loss of minor grooves. Is it clear? So, when it reaches the melting temperature, double stranded DNA becomes single stranded and instantly the signal drops to 0. So, this is one of the criteria to judge the quality of your primers. Okay. If too many mispriming, you will get too many melting peaks. You need a single melting peak if it is a successful real time PCR. Change in fluorescence when double stranded DNA incorporated with incorporated dye molecule melt into single stranded DNA as the temperature raised. Did you understand what happens here? The double stranded DNA, when it is subjected to a specific temperature at which double stranded DNA becomes single stranded, instantly the minor groove is lost, the DNA, the dye which is bound in the minor groove is detached. So, there is a drop in signal. Okay. Yeah, that is what just now I explained. Double stranded bound cyber green dye is heated, a sudden decrease in the fluorescence is detected at Tm. Tm stands for melting temperature. Yeah, this is the change in fluorescence and change in temperature is plotted against the temperature to obtain the clear view of melting dynamics. Okay. Yeah, this is one of the applications. I already told there are two major applications of real time PCR in our pathology and microbiology. One is absolute quantitation, another one is relative quantitation. Okay. The level of expressed gene may be measured by absolute or relative quantitation in real time PCR. Absolute quantitation relates the PCR signal to input copy number using a calibration curve, while relative quantitation measures the relative change in mRNA expression level. We will have a detailed discussion in the practical class. Whatever doubts you have, please write down and we will have a discussion there. And there are two basic types of real time PCR. One is absolute and relative. The choice is based on your experimental need and your experimental setup. Some more points about absolute quantitation. In absolute quantitation, you are estimating the number of templates copies in your sample. So, it requires a construction of absolute standard curve for each target gene, and the standard curve is based on the serial dilution of the sample with a known copy number, and CT value of each standard sample is plotted against a logarithm of the known concentration, and the standard curve is then used to estimate the concentration of unknown samples. So, we are going to demonstrate that. That will give you much better clear view of how to do this absolute quantitation. This is relative quantitation. Changes in the gene expression in a given sample relative to another reference gene is analyzed. Calculation methods for relative quantitation, there are different methods are there, and we need to do certain activities like normalization, and of course you have to use a reference gene. Remember, basically you have to use, I will take you that particular point, so forget about that. This is very, very important. In relative quantitation, which most of you are quite familiar, always remember, in relative quantitation, you have to use two different control points. Okay, one is called as calibrator, another one is called as reference. Right? Let us take small example that will give you a better clarity on this. Calibrator is nothing but a plant which is not treated at all. Okay, in all your pathology experiments and microbiology experiments, what are you going to do? In pathology experiments, you will be inoculating the plant with a pathogen. In microbiology, you will be inoculating the plant with microorganisms. And necessarily, you have to keep one control, which is not inoculated with the pathogen or not inoculated with the microbe. So, that is called as calibrator. And reference, and this is your main, that is the test plant. Okay. Example wise, you have mock plant, housekeeping genes, this is the inoculated plant, and what is called as the main target and effector gene. We will have small discussion once again in the classroom how to do this. Take it as a message, we have three common approaches for relative quantitation. One is Levac method or double delta CT method, another one is the delta C method using a reference gene, and beef apple method. These three are the most common method in which you can use, calculate the relative expression of your genes in your experiments. Okay. We will demonstrate, I think, double delta C method in our lab, in our practicals. We will do that. That is going to be the last one. Of course, these are all some of the applications. We have gene expression analysis, detection of mutant genes, copy number, pathogen microbe detections, and of course, single nucleotide polymorphism, and many more promises are there in real-time PCR. I think this is the last but one slide, and the number of companies are available in the market who manufactures and distribute this machine. We have from Roche, we have ABI, we have Bio-Rad, Strategenes, and so many other machines are there. And the one machine we are going to see tomorrow, that is from Roche 96 light cycler. With that, I will conclude. The second part of the lecture, that is more of a practical in nature, that will have a detailed discussion in the practical room. How to do absolute quantitation, how to do relative quantitation, and the formula which is needed to do the absolute quantitation, and the programs which is online available for relative quantitation. All those things we will demonstrate in practical. Okay. So, the primary purpose of this lecture is let you know the fundamental differences between dye based real time PCR and probe based real time PCR, and the principle behind this dye based real time PCR and the probe based real time PCR. Okay. With that, we will stop the morning lecture today, and we will break for tea, then come back. The next speaker has already arrived. So, we take 5 to 10 minutes maximum. 11:40. Okay. 11:40 we will start the second lecture. Okay.